Bismillah Rahman Rahim. Welcome back, guys. Welcome to another video sessions from Kame Biology. And in this video lecture, we will talk about the life cycle of the herpes simplex virus. Now, what is herpes simplex virus? Herpes simplex virus are those viruses which can cause herpes viruses. Fine. So now let me write the life cycle of herpes viruses. In short form, we write HSV means herpes. H means herpes, S means simplex, and V means virus. Herpes simplex virus. Herpes simplex virus are the non-enveloped virus. They can cause herpes inside the human body. Now, what is herpes? Herpes may be in two forms. One we call that herpes simplex virus 1, and second we call it herpes simplex virus 2. In first case, in herpes simplex virus 1, they can cause infection in the skin around the mouth. What happen in this herpes simplex virus 1? They will cause the formation of blister watery. Means they can produce some abscess around the mouth or on the face. These abscesses are full with pus and these are full with water. So that's why we can call that water blistery. And they can also cause itchy, means they can cause itching and also cause the irritation as well. These abscesses are very painful. Fine. So they can cause these kind of abscesses. In second case, herpes simplex virus 2, in this case what happen, they can cause water blister, or they can cause the production of abscess on the reproductive organ, means on the genital area, both in male and in female, by which that area will be irritated or will be itchy, means itchy will also occur. So that's all about the herpes simplex virus 2. Fine. So these are the disease which are caused from the herpes virus. So now let me write, it can cause herpes. Fine. Now, herpes simplex virus are the enveloped virus. They contain double-stranded DNA, and there are also some special and necessary proteins we call the tegument proteins. And there are also the spikes, means the spikes will also arise. We call it which are made up of glycoproteins. Fine. So how it will be look like? It will be look like. Let me draw. This is the capsid. Inside the capsid, there are the genetic material, but the genetic material in this case is the double-stranded DNA, and around the capsid there are envelope. So they contain the envelope. Now between the envelope and the capsid, there are some special proteins, and we call that tegument proteins. These black dots, not only black dots, this blue one also. Now these blue and black one represent the tegument proteins, but these tegument proteins are actually two. The blue one, let's suppose the blue one are called that virion host shutoff, means VHS, virion host shutoff. And the second one, the black one, as I draw here, these are called VP16. And collectively we call that what? We call that tegument protein. Tegument protein. Now the tegument protein function we'll be talking later. Now on the envelope area there are also spikes protein arises. But the spike protein in this case are 4 different spike proteins. Let me draw one off with the green, and this one off with blue, because it contains 4 different spikes protein. And let me draw this one with red. Yes, okay, fine. Now after that, what happened, these 4 are actually the receptors, or not only 4, but majorly these 4 are the important receptors which are repeated again and again and make a spikes proteins. This green one, let's suppose this green one we call that GD. This blue one, let's suppose we call that GH or GL. This black one, let's suppose we call that GB. And this red one we call that GB. And collectively we called all these called that surface glycoprotein. Surface glycoprotein or receptor, surface receptors. Now let me explain the glycoprotein in a short form. That what happened on the host cell, there are also receptors available, but these receptors which are present GB, GD, GB, GH and GL, once the host cell recognize any one of them receptor, so they will be attached and then they will be internalized. Fine. So the host cell just recognized in each of them, the host cell receptor will just recognize one of the receptors. Fine. So then they will be internalized. What happened, this virus will present on the host cell and they will circulate, they will move just like a circular form, and once they will attached on the host cell receptor, or once the host cell receptor recognize any one of them glycoproteins, so then they will be internalized. Let's suppose this is the host cell membrane. Fine. So here are the host cell receptor. So what happened, this black one, let's suppose recognize this black one receptor and make a covalent interaction here also. So let me draw the structure here, the virus, the herpes virus structure here in a rough form. So let's suppose this is a virus in a rough form. Let's suppose the genetic material are also present. Tegument protein are also present. I will draw here after that. These are the tegument proteins as I dotted. So what happened, once they will attached, then they will be internalized through a membrane fusion. Why? Because this virus is the envelope virus. So they will be internalized through a membrane fusion. So how, when they are internalized through a membrane fusion, how they will be look like? They will be look like just like in this form. The envelope area will remain outside, while just the capsid and the tegument protein, means the capsid area and the tegument protein will be remain inside form. These are the double-stranded DNA and also the tegument protein. So these are the tegument proteins. Now what happened? Once they will internalize inside the host cell, then this virus will be moves inside the microtubules. So now let me write here, moves inside the microtubules and then reach to the nucleus. So let me draw the nucleus structure here. Let's suppose this blue one represents the nucleus structure, nuclear membrane, or we can say. So the virus once reached here, so they will just leave their capsid. These are the capsid. They will leave the capsid and just the DNA will be internalized. Just the DNA will be internalized. What happened? This capsid portion will remain outside and the DNA portion will be internalized. As I told you that there are two segment proteins. One is VHS and the other is VP16. Now what are the function of these two proteins? The VSH will degrade the host messenger RNA. Let's suppose this green one represents the host messenger RNA. The host messenger RNA are also present. So what happened? These VSH will goes there and they will start degrading, or they will finish the function, or they will start degrading the host mRNA. These are the mRNA. Fine. And what are the function of VP16? What are the function of this? This VP16 is act like a RNA polymerase. We can also call it. It's act like RNA polymerase. Why we call that RNA polymerase? Because it will sit on this genome and produce messenger RNA, or transcription process will occur and we get the messenger RNA, early messenger RNA. This red one represents the early messenger RNA of what? The early messenger RNA of virus. Early. Fine. And they will be released out to the cytoplasm. Let's suppose this is the early messenger RNA, and this early messenger RNA will start the then after that the translation process will occur, and then we get what? We get the proteins. But these proteins are called as early products, or we call it early proteins. These early proteins and messenger RNA will again take inside the host cell. So they will be again taken inside the host nucleus. Once they will reach inside the host nucleus, so this proteins, as I draw early proteins, now these are used to produce DNA, double helix, double stranded DNA they will produce. So they will take this RNA as a template, and then we get what? We get duplicate or many structure of the double stranded DNA. Fine. Let's suppose many structure of DNA we get. After we get double stranded DNA, then what happen? Then this DNA will produce late mRNA. Let's suppose this black one represents late mRNA. This late mRNA are again released out from the host cell, from the host nucleus, and produce what? Produce the structural proteins. Now these blue ones represent the structural proteins. Again, these structure protein and mRNA are again means they will again go to the inside the nucleus, And what happened, the structural protein and the DNA will around and will combine together, and we get the capsid plus double stranded DNA. So we get just naked portion of the virus. So how it will be look like? So it will be look like just like that. Let's suppose this blue one area represents the nucleus, and here the virus, not complete virus, just the capsid portion and the nucleic acid will form. Fine. Now this will, whenever they are released out from the nucleus, so how it will be look like? It will be look like just like that. So they will take some portion of the nucleus, means they will be come out from the nucleus to taking some portion of the nucleus. And how it will be look like? It will be look like just like that. Here is the nucleate portion, and then after some time they will be start degrading, and then once they will release out from the nucleus, then they will reach to the endoplasmic reticulum. Now inside the endoplasmic reticulum, or outside or inside the endoplasmic reticulum, the protein are also formed. It means the receptor proteins are available here. So the virus will get the proteins here. So the herpes simplex virus will get the receptor proteins and the envelope area here. So once the virus will enter here and then release out, and after that the virus will also passes through from the Golgi bodies. Let's suppose this is the Golgi body. So the virus will also passes from the Golgi bodies, and when the virus are coming out from the Golgi bodies, so now we get this structure of virus. What does it mean? It means that we get a complete, or we get a mature and well virus. Means the complete virus as required. The complete virus are now formed. So we call that progeny virus. And then they will be released from the cell through a membrane, budding, through cell budding. Sorry, not membrane budding, through cell budding. So they will be released through cell budding. And once they will release through cell budding, they also take some portion of the cell membrane, host cell membrane. They also take some portion of the cell membrane. And after that, then they will are ready to attack another host cell and start the life cycle again and again. And finally, what they cause infection, and after the infection they cause the diseases. Fine. So that's how the HSV, means herpes simplex virus, can start their life cycle and can do their, that how they can start or can do their life cycle. Fine. I hope you understand. If you like the video, please hit the like button, share this video, and also subscribe my channel to get more interesting video lecture like that. Thank you so much.